Hello, and welcome to my oral presentation for the term project for Biology 3550. For this project, I was assigned sequence number 22, and the first thing I did was complete a BLAST-N search and found that my sequence is likely from the genome for the cauliflower mosaic virus. I was then able to identify 5 closely related sequences and one a little bit more distant related sequence, and I was able to run those through a BLAST-P analysis, and also I was able to then find the translated sequence with the ORF finder, and I was able to identify the open reading frames in the sequence. The translated sequence was then used to identify the motifs. I was only able to find one motif, and it was located at approximately the 500 nucleotide mark. No peptide secretion signals were identified, and my sequence also didn't contain any transmembrane domains. Signal P was used to predict the peptide secretion signals, and TMHMM was used to find the transmembrane domains. This is my cladogram made by Clustal Omega with the my sequence and then the 5 closely related sequences and then the one that was a little bit more distantly related. I found that the closest relative appeared to be the binary vector PLX-CAMV. I then ran a fire analysis and found that the molecule that I got was the ribonuclease H1, and the title that it gave me was the hybrid binding domain of human RNase H1 in complex with the 12-2 mer RNA DNA. However, it wasn't a super close match. It only contained 43 residues, which is 8% of sequence 22. My conclusion is that sequence 22 is most likely from the inclusion body matrix protein located on the cauliflower mosaic virus, and the closest relative was the binary vector. And this concludes my oral presentation. Thank you so much for listening, and I apologize about the formatting.